Today we'll be discussing Chapter 13, which is Immunodeficiency. So, what is immunodeficiency? It essentially is a defect in the immune system that leads to susceptibility to infection. And it can actually, as we'll talk about, also involve autoimmunity due to regulatory defects in the immune system. So there are two types of immunodeficiency. The first is primary immunodeficiency, which is due to a genetic defect in a portion of the immune system. The second type is secondary or acquired immunodeficiency, and this is a non-genetic defect that is acquired during life that leads to susceptibility to infection. So why study immunodeficiency? First of all, genetic defects in immunodeficiency are very rare. They occur in very small portions of the population. However, these defects help us really understand the mechanisms behind the immune system, and especially how different parts of the immune system interact. And these defects are especially important to help us understand human immunology, since we lack many of the tools that we have in experimental animal models. So there are many types of primary immunodeficiencies. In fact, there are over 150 different immunodeficiencies that have been described. And they often first show as severe and recurrent infections during childhood. And as I mentioned before, these defects, when we look at them together and see the symptoms that they cause, really help us understand the molecular basis of the immune system. So, not all genetic defects in the immune system actually lead to immunosuppression. We'll talk about this a little bit later, but this is important because we can see which areas in the immune system are redundant. So if you lose one, it's covered by another portion of the immune system. So we're going to be talking about several defects, and the Janeway textbook organizes these into T-cell defects, which will talk primarily about SCID, and B-cell defects, which we'll talk about as well, which these typically involve antibody changes. Then there are a few other miscellaneous genetic defects that we'll see. So, although the defects are organized this way in the book, this is not necessarily the most accurate way to describe these diseases. And that's because, as you'll see, the different portions of the immune system, such as B cells, will actually interact with and affect other portions of the immune system, like T cells. So there are a few things to keep in mind. We're going to be going over a pretty big number of diseases, and it's not important that you memorize the list of diseases and their symptoms. What's really important is that you deeply understand the way that we can use these specific defects to understand immunodeficiency. And understand as well how the symptoms of these immunodeficiencies lead to the symptoms that are seen in patients. So we'll primarily be talking about human immunology in this lecture, and we'll be talking about and reviewing many of the concepts that you've covered in previous lectures. So we'll start with T-cell defects, as they're organized in the book. So the first is SCID, or severe combined immunodeficiency. And this is sometimes called the boy in the bubble disease because there has historically been patients that have this disease and they fully lack adaptive immunity, and therefore they live in a bubble so they can avoid infection. The most common type of SCID is X-linked SCID. And patients here typically have absolutely no function of T-cell or B-cell immunity. Essentially, they lack an adaptive immune system. So I want you guys to think about what possible genetic defects could lead to this. And how do T-cell defects in particular lead to SCID? So first we'll talk about X-linked SCID. And we can see in these patients that they have no normal T cell or NK cell function. And they also have a decreased T-dependent B cell response. So these things initially are pointing to a defect in T cells. So how can we determine what genetic defect is leading to this disease? So we have some data on other immunodeficiencies that we can use to help us. So first, we know that human IL-7 receptor alpha is a specific defect where patients have no T cells but still have NK cell function. And this is unlike our X-linked SCID, where patients lack both T cell and NK cell function. We've also seen in mouse experiments that IL-15 mutations have no NK cell function, but they do have normal T cells. They also have a defect in CD8 T cell memory. Okay, so with this information, and thinking back to the things we know about T cells and the receptors, particularly the IL-2 receptor gamma C subunit, we can determine that X-linked SCID is due to a defect in this subunit. And if we look back at our data, we see that defects in IL-7 receptor alpha and IL-15 show some of the phenotypes of the X-linked SCID. And this makes sense because the IL-2 receptor gamma C subunit is a subunit of the receptors for IL-2, IL-4, IL-7, IL-9, IL-15, and IL-21. So this is just a first example showing how we can use data on genetic defects to better understand the immune system and the genetic cause of a disease. Okay, so there are other multiple different types of SCID. So X-linked SCID is the most common, but these defects also lead to a lack of functional T and B cell immunity. So first of all, we can think of a RAG1 or RAG2 mutation. So this prevents DNA rearrangement and VDJ recombination. So without this, we don't get any mature T and B cells. We also have two defects that lead up to a buildup of toxins, which is lethal to T and B cells. And another defect that's similar to the RAG1 and RAG2 is the DNA double-strand break repair defect. And this is important because after VDJ recombination, DNA double-strand break repair is needed to put the DNA back together. So without this, the T cells cannot progress. So we can see in this figure, this is figure 13.2 in the book, if you want to see in more detail. So we'll first look at the top half, which is T cell defects, and the bottom half, which we'll talk about a little bit more later, is B cell defects. So a couple that we talked about are here, the IL-7 receptor alpha defect and the RAG1 and RAG2 defect. And both of these defects lead to SCID. And we can see why that is because of their position in the T cell maturation cascade. So they're especially important in moving between immature T cells and pre-T cells. So if this step is interrupted, the T cell development cannot continue. We can also see that there is SCID here in the B cell area, which is seen, it's our ADA defect shown here. So that affects primarily B cells, but it's also important for T cell development. Okay, so from this we can see that B and T cell development are very linked. There's a lot of crossover. And defects in one can lead to defects in another. So the groupings, as I said before in the book, are not entirely accurate, but they give us a general idea of a way to categorize these diseases. So it's also important, since I just showed that graph of T and B cell maturation, it would be really good if you guys went back and reviewed your lectures on B and T cell development. That'll give you a good review of ways that genetic defects can lead to specific diseases. So again, this is just an overview of that. And we can also see a few other defects leading to SCID. There's the ADA defect, as I pointed out, in B cells here. And there's also a few other genetic defects a little later in the T cell development which cause SCID as well. Okay, so our next disease that we're going to talk about is Wiscott-Aldrich syndrome. So let's say we have a patient that comes into the clinic and presents with several symptoms. So these symptoms are blood clotting problems and low platelet numbers, an autoimmune susceptibility, as well as susceptibility to recurrent bacterial infections. We also see that this is more common in male patients. So, what genetic defects could possibly explain this cluster of symptoms? So, through genetic analysis, scientists have discovered that this disease is caused by a defect in the Wiscott-Aldrich syndrome protein, or WASP. So the WASP defect leads to problems with T and B cell synapse formation, and as well as Treg development. So what WASP does is it actually helps control actin polymerization in the cells. So when the immune synapse is created, the cell must contract, and therefore the cell needs functional actin. So without the WASP protein, this contraction cannot occur, and T and B cells cannot fully develop. And we also see, since this is important for Tregs, that explains the susceptibility to autoimmune diseases as well. Okay, so because this is more common in males, we can assume that this is a X-linked recessive genetic defect, which would be verified in experimental genetic analysis. And we can also see that the WASP protein, as I showed in the last slide, is expressed in hematopoietic cells. I'm going to start that sentence over. Okay, so we can also see that the expression of WASP in hematopoietic cells controls actin polymerization. And since immune cells as well as blood cells are developing in the bone marrow, they are affected by the WASP defect. So this also helps explain the blood clotting problems and low platelet numbers. So, as I said, this disease, when we see the protein defect, has an effect on both lymphocytes and platelets. And so this is a really good example of how scientists and physicians can use symptoms and what we know about the immune system to help understand how defects in specific proteins can cause problems, therefore giving us information about how the immune system works. Okay, so another category of deficiencies is Mhc deficiencies. And here we'll really see the important role of protein interactions and transcription factors. So we'll first talk about Mhc-2 deficiency. So as you guys talked about in previous lectures, CD4 T cells interact with MHC2. So if you have problems with your MHC2, you're going to have fewer CD4 T cells. And that's exactly what we see in these patients. They have normal CD8 T cells and no functional CD4 T cells. There's also a decrease in the T cell-mediated antibody response, which when we think about how T and B cells interact, we know that CD4 T helper cells are important for the development of antibodies, and so that helps explain this symptom that we see. And interestingly, the MHC defect is actually not a defect specifically in the MHC2 protein itself. It's actually a defect in the proteins that are required for transcriptional activation of MHC2 genes. So this is actually a defect in the transcription factor that leads to MHC2 expression. So without that transcription factor, we do not have MHC2, leading to these defects. So now we'll talk about Mhc-1 deficiency. So the opposite of MHC2, we see no normal CD8 T cells, while the patients have normal CD4 T cells. And these patients are also susceptible to chronic respiratory infections as well as skin ulceration. And this is due to a TAP1 and TAP2 deficiency, which, if you'll remember, if you go back to the lectures on MHC and T cells, this is a protein that's important for peptide loading into the Mhc-1 complex. So our next category of deficiencies we'll broadly call B-cell defects. So B-cell defects in general have a set of common symptoms, and this is due typically to a decrease in antibody response. And this antibody response defect leads to an inability to clear extracellular bacteria and viruses. And this is especially true of pyogenic bacteria such as staph or strep, as these are not recognized by phagocytes. And also, B-cell immunodeficiencies lead to specific susceptibility to enteroviruses, which is important for gut infections, and antibodies really help control these enterovirus infections. So our first genetic defect that we'll talk about under the B-cell defect category is X-linked A-gamma globulinemia. And so A-gamma globulinemia, if you break the word apart, means a lack of gamma globulins. And this is actually not typically detected until several months after birth. And that's because a few months after birth, maternal antibodies start to decline. And if the patient lacks gamma globulins and antibodies, then when the maternal antibodies decline, they are left without functional antibodies. And this is due to a mutation in BTK, which we'll show in the B-cell development pathway a little later. So BTK is a tyrosine kinase, and it's really important for pre-B-cell receptor signaling. And so if this signaling is disrupted, then B-cells cannot mature. And in female carriers, interestingly, all B-cells actually are functional. And this is due to X inactivation. So here you can see on this same pathway, we can see where BTK is important. And so if BTK is not functional, the B cells cannot progress. So I mentioned that female carriers actually have completely functional B cells. And the reason for this is due to X inactivation. So in males, a healthy male without a BTK defect will have proper development, and the B cell will be able to mature. However, if they have a copy of the BTK deficient protein, then these B cells will have arrested development and they will not continue to mature B cells. However, in females who have two X chromosomes, if they have one functional copy and one non-functional copy, all the B cells that express the functional copy will mature and become functional B cells. However, all the B cells that express the non-functional copy will have development arrested. So this leads to the entirety of the B cells all expressing functional B cell receptors, because the ones that have the dysfunctional BTK will not progress in development. So our next B cell defect is called hyper-Igm syndrome. So in hyper-Igm syndrome, we actually have normal B and T cell development, but we see very low antibody titers. And in fact, we only have IgM present. And this leaves individuals susceptible to extracellular pathogens. So in this disease, we see mature B cells, but B cells cannot class switch beyond IgM. And interestingly, this is not due to a defect in B cells, but actually a genetic defect in T cell help. So without functional T cell help, we have no class switching or somatic hypermutation. And you can really see the effects of this in these histology diagrams, where in the top, in a lymph node from a patient with hyper-Igm syndrome, we see no germinal centers. So there are no sites where class switching and somatic hypermutation are occurring, and this is compared to a healthy lymph node. Okay, so this again is due to T helper cell defects, which leads to B cell defects. And we can also have defects here in some proteins that cause interactions between T and B cells. So for example, CD40 and CD40 ligand would be expressed on T cells and B cells respectively, and as well as T cell and dendritic cells. So these interactions would then be disrupted, and this can lead to a lack of T cell help being provided. The next set of immunodeficiencies are called common variable immunodeficiencies. And this is a group of related mild immunodeficiencies, and they're mild enough that they often are not diagnosed until adulthood. They typically involve defects in a single antibody isotype. For example, a defect in co-stimulation for B cell activation and class switching would cause problems with this. So these patients may have mild susceptibility to certain infections, or it's possible that they have no symptoms at all depending upon the defect they see.